So thank you. It's a great honor to be here. PrevEP stands for Prevent Epilepsy. We founded the company in 2018 to develop treatment to prevent epilepsy after acquired injury. What Indiana Jones is holding here is the Holy Grail. The Holy Grail of epilepsy is finding treatment to prevent it. Epilepsy causes include acute CNS injury, such as head injury, stroke, and infection. With all these, epilepsy is potentially preventable, but we have no treatment to prevent it. And I'm here today to ask you to help us develop it. There have been two obstacles to development of treatment to prevent epilepsy. One, there has been no treatment, pretty important. But two, the logistics of doing clinical trials to prevent epilepsy after head trauma are substantial. We think that we have solutions to both of these problems. So our co-founder and CSO, Wolfgang Glöscher, has developed a concept of network pharmacology for epilepsy prevention, in which drugs that target different aspects of the disease are used together with a greater effect than if if a single drug targeting a single disease mechanism were used alone. He has used this concept to develop combination treatment using approved repurposed drugs with known anti-epileptogenic effect in animals and a good side effect profile. He evaluated 500 compounds in different combinations and came up with 12 leading combinations that he tested in the commonest anti-epileptogenic model in epileptogenic model in animals, the rodent status epilepticus model. 3 were beneficial, including one, PrevEP 002, which you can see here, which completely prevented epilepsy. PrevEP 002 consists of ceftriaxone, atorvastatin, and levetiracetam. Between the 3 of them, they reduce glutamatergic toxicity after injury, they reduce inflammation, and they modulate neurotransmitter release. We're now further characterizing these findings in an NIH-funded study of TPI model of PT in rodents in the lab of Alex Rotenberg, a co-founder of PrevEP at Harvard Medical School. And as part of the same grant, we're also developing all the other things that we need to do to go clinical, including an FDA meeting, which we hope to have in about 18 months' time. So we think that we've solved the treatment problem. How about the logistics? There are 3 main problems. One, the sample size needs to be large because not all patients with TBI develop post-traumatic epilepsy. 2, you've got the issue of feasibility of enrollment of patients. And 3, you've got a major problem of long duration of studies because of latency between injury and epilepsy. We think that we solved the first 2 problems. By using imaging biomarkers, we've come up with identifying patients with 30% risk of post-traumatic epilepsy. And with 30% risk, you only need about 320 patients to show efficacy if you're going to reduce epilepsy by 50%. We've also showed that we can enroll these patients, as we have done in a DoD-funded study of biomarkers of post-traumatic epilepsy prediction. However, one problem remains, and that's the long duration of the study. So about 80% of all epilepsy after head injury starts within 2 years. And for that reason, that has been the duration of studies in the past. And that is too long a period of time for a proof of concept study. And without a proof of concept study, we will have a very hard time funding the funds to fund the phase 3 study to show whether PrevEP 002 can prevent epilepsy. So this is where we come to you, sharks. We need a biomarker of early post-traumatic epilepsy detection. Early after TBI, you can have seizures, and those seizures are more commonly present on EEG than clinically. So we think that an EEG subclinical seizure may be a biomarker of early post-traumatic seizure epilepsy detection. And so here is our proposal. We propose to do a pilot study to develop a biomarker of early post-traumatic epilepsy detection by using long-term continuous EEG monitoring and a subclinical or EEG seizure as the outcome. We will use the award-winning UnEEG that you heard about before that can do ultra-long recording. We will start the recording shortly after TBI, and we expect that EEG seizures will precede clinical seizures to give us that biomarker. Successful pilot will allow identification of post-traumatic epilepsy early, will allow us to do a proof of concept study of efficacy, and that will then allow us to move to phase 3 study. This is our team. And with that, I'd like to thank you all for your attention. And if I may, when you vote, I'd like you to remember 3 things, if I may, please. 1, prevention of epilepsy will make a huge difference in patients' lives. 2, PrevEP has a promising treatment to prevent it. And 3, and this is the most important one, you sharks can help us to do it. Thank you.
Pavel, nice job. And I think certainly agree that the ideas behind this are really important, right? And this model certainly is a way to look at epileptogenesis, right, from after an insult. I have a few specific questions. One, I'm interested, can you tell us a little bit more about what your radiological markers are that make it a better predictor of this outcome?
So historically, so historically, the studies that have been done that really have been the best done studies use patients with a 20% risk of PT within 2 years. Those are studies that are done by Nancy Tamkin, and we did a similar study ourselves. When you look at certain neuroimaging markers, patients have a much higher risk of PT than that. And these markers include 1, subdural hematoma with surgery. That's not just an imaging marker, but it's 2, multifocal parenchymal hemorrhages. Doesn't matter where they are. The Epibar S4X study shows that bitemporal are good, but it really doesn't matter where they are. 3, depressed skull fracture, and 4, combination of subdural and parenchymal hemorrhages. These 4, between them, have an overall risk of about 30%. Now, we've just completed enrollment into a DoD study of PT biomarkers using these criteria, and we found that we could enroll. And we did it during COVID, and we were able to enroll during COVID.
Okay, very good. We got one more since I'm going quickly. Okay, fine. Yeah.
So I'm curious, you talk about monitoring people EEG wise prior to them having seizures. At what point in time do we start the monitoring? And then do PLEDs, which is a sign of acute brain injury, influence your choice in terms of, will this person go on to develop post-traumatic epilepsy or not?
That's a great question. So acute monitoring after TBI is not new. It's been done. It has been evaluated very well by the Epibar S4X study that Brandy helped to initiate, that has looked at continuous EEG monitoring within the first week after trauma. Let me take a step back. For reasons that have to do with epidemiology, seizures after TBI are divided into early seizures, which don't necessarily lead to epilepsy. They have an increased risk of epilepsy, about 25%, but they're not necessarily epilepsy. Whereas unprovoked seizures that happen 7 days or later after TBI automatically lead to, in 90% of cases, lead to further seizures and are called post-traumatic epilepsy. So you're right about the acute changes. We will plan to implant within the first day of injury, and then monitor for up to 6 months. We're not so interested in the first week because that work has been done or is being done. What we're really interested in is the seizures after the first week, and specifically whether they happen without clinical seizures.
Yeah, I'll just follow up a little bit on that. So with the UNEEG system, you're recording from a small portion of the brain, and how, with these multifocal injuries in the patients you'll be recruiting for the study, how can you ensure you'll either put it in the right place? And then the second question and follow-up is, to use this new 3-drug combo, you really need a biomarker before the epilepsy starts. And wouldn't a electrographic seizure seen on the UNEEG be too late?
So the first question is a really good question, and we are limited by the UNG's spatial resolution. There is one group of patients that will lend themselves or set of patients that will lend themselves well to the UNEEG. Number one, subdural that requires surgery, which is unilateral. Number two, a subdural with parenchymal hemorrhages that are more prevalent on one side. And number three, if you've got a weighting to the multifocal hemorrhages that is one-sided. But your point is well taken. It'd be great if your UNEEG developed bilateral monitoring system so we could do it on both sides. The second question is we don't really need a biomarker of the well, let me step back. The goal with the UNEEG is to record the seizures EEG-wise before they get manifested clinically. So let us say that a patient has a first seizure at 9 months. The assumption is that they've been seizing before then, but we haven't seen it. And if we can detect that seizure at 2 months, then we can do the proof of concept study to show whether or not there's a signal of efficacy. That will not be a biomarker that the FDA will accept for the pivotal phase 3 study, but it will allow us to move forward to the phase 3 study. Does that answer the question?
Yes, thank you.
I like that one. But, let's talk about money.
The more the better.
You have my vote. No, not yet. Commercialization, I'm not clear. Is the biomarker or the 002 combination your product?
The product is the 002 combination, and the goal is prevention of epilepsy. What the biomarker is answering a critical bottleneck in the clinical development, moving from the phase 1 pharmacokinetics to phase 3 and not having the funding for phase 3 without the investors seeing there is a signal.
Okay, and then my understanding for the product is the plan is to charge for the cost avoidance and so of treatment by preventing the epilepsy, and that fee was going to be approximately $60,000, give or take, if I recall. The question I have is the product is made from 3 readily available molecules, and in creating it, you're going to necessarily have to disclose what that combination of those 3 molecules is. So in a sense, it's like you've discovered the Holy Grail, but it turns out it's like a plastic cup at Whole Foods. What's to prevent everybody from just putting these things together and, you know, selling them themselves or just ordering it for your patient without paying that fee?
So I think Indiana Jones would disagree. It's not a plastic cup. And the reason is the following. You're absolutely right. Anybody could take those 3 medications and combine them. Not so for IV. So our IP protection is not only for the concept, which I think you're right, could be bypassed, but for the IV formulation, which cannot be. So ceftriaxone and levetiracetam are IV available, atorvastatin is not. And we found a way of formulating it that is patent protected.
Sounds like you need me.
Questions? Good. Okay, thank you, Pavel.
Thank you.